Consortium. And today we have the pleasure to receive Aled Edwards. He's already a visitor professor in our center and comes here pretty often. But today he'll tell us about how he really thinks the models that use open science are expanding outside academia to pharma companies. So Aled is a professor at the University of Toronto and some other universities where he's a visitor. And he has started many companies at the beginning of his career, but at some point, 20 years ago, he thought that we needed a different model where he participated in the very early steps of open science for partnerships between academia and industry. And basically, this is what you tell us a little bit about today. And at the end of the seminar, right, so you guys can ask questions or you can stay after and talk to him if you wish. Okay?
Thank you. Do you want me to use? Okay. Okay. So I didn't put up the, hi there. I didn't put up the presentation because I want to show you, I have a new grandson. It's our first grandson, and this is what he thinks of Trump. All right. Luckily, we live in Canada and we don't have to deal with Trump. There we go. So the question is today, how can we use different styles of doing science to try and have an impact on society, and specifically the invention of new medicines. And so the first part of the talk, I will start to speak about what is the problem in the system. And so, around the world every year, we society, about $300 billion in biomedical research and drug discovery. About $150 billion by governments and foundations and charities, and $150 billion by industry. So in total, we have trying to uncover, how life works and to try and make medicine. Disease that he lived in 1808. So 200. That's when he described Parkinson's disease. So if you think we've 200 years now, and if you go to a doctor, a neurologist, and you ask, it's the system's fault, right? The system is stupid. So can we start sharing more and use the $300 billion to do more research per dollar? That's the hypothesis, okay? How do we expect to cure these diseases if we don't know what causes them? Similarly, Alzheimer's lived 100 years ago, and we still don't know what the cause of Alzheimer's is, let alone how to treat Alzheimer's. So, as scientists like us in the room, we're doing a very poor job of trying to explain biology. And so, why are we making new medicines? We have to sort of look in the mirror. And moreover, when we do make new medicines, they're too expensive for most people on the planet, right? Because the investments required, the failures of the industry to successfully predict which drug is going to work. It's not their fault, it's our fault. We're professors, we're supposed to tell them how Alzheimer's works. They're supposed to cure Alzheimer's. We don't tell them how Alzheimer's works, so they guess. And they guess wrong most of the time. And when they guess right, they have to charge a lot of money to make up, that's what they say anyway, to make up the price of how much they spent on the failures. Okay? And so we have a system that really doesn't work, right? We don't discover biology well, and when we do make medicines, they charge too much for most people. That's a big problem. So that's the problem we like to tackle. So let's now look at professors, and I've showed this slide before. Okay? We spend $150 billion. I argue we only really get $20 billion in value. And to show this, it's an experiment that we did, a library experiment. And so I took the year 2016, and on this graph are the 512 human kinases, and on this, how many papers on each kinase. So these are the number of papers, because for every kinase has a name. Let's say it's SARC, you type SARC into PubMed, it says there are 3,422 papers on SARC. You just go, Okay, and then you go through every single one of the kinases, and this is the graph you get. So you can see that a lot of the kinases up here get many papers in 2016. But the order of these kinases is not by alphabetical. The order is how popular they were, how many papers there were in the 1900s. And so if I was to take all the kinases and rank them by how popular, the most papers to the fewest papers, prior to the year 2000, that's the order. So what does that say? That in 2016, most of the scientific community still works on the kinases that were popular when I was starting my research career. So you ask, well, why aren't they curing diseases? Well, maybe it's because we're not looking everywhere. We're just looking at the same things we always looked at. Okay? And it gets even worse. And so I made the joke before that there's one thing about Canadians that everybody knows, that we're smarter than everybody. And so let's now do this exact same search in PubMed and go SARC plus Canada, which will tell me only the papers in the world that were published by Canadians or with Canadian co-authors. Okay? And so let's look. And if I did SARC and Brazil, I get the same pattern. So we take $150 billion around the world, we divide it into countries. FAPESP only funds Brazil, Canada government only funds Canada. We read the grants are always the same, everybody's doing the same research all over the world, and then you get this, the same. Okay, so it's a really poor use of global money by academia. And remember, every time we study one of these genes, we're not studying one of these. And it keeps going like this. So we keep studying the same thing, and we're not getting any cures. And you wonder, maybe we should think of something. So we're getting a poor investment for our global investment. And in addition to that, when we started the SGC, we say, this is where the fund is going to be, on the proteins that nobody knows about. It's very difficult to get funding from FAPESP or from Canada to fund one of these genes because there is no hypothesis. In a FAPESP grant, you'll say this protein is important because the mouse does this, and I do this, and I knock it out, and it interacts with this, and I hypothesize this, and they'll say, yeah. Well, you can't write that about one of these down here. You can only write good grants on proteins that are up here, where there's a lot known, so you can write a grant that makes you look smart. But the smart experiment is actually to study these ones, not these ones over and over again. Industry has the same problem. So what industry does is it takes one of these ideas and says, I want to make a medicine because I think inhibiting protein kinase X will cure cancer. So they start a drug discovery project. They start with the kinase, they make a chemical, they proceed the chemical down until they test it in the cancer patients, and usually this fails. But just like every professor works on the same thing, every company works on the same thing. And so for Aurora kinase B, this fails in Pfizer, and it fails in AstraZeneca, and it fails in Novartis, and it fails in Merck, and it fails in Takeda, and how much money is being spent? This costs about, I would say, $50 million, right? 7 years, fail, fail, fail. And what's even worse is that these data aren't published. So when the first one fails, the second one doesn't know, keeps failing. You hear a rumor, but you don't really know. And so we waste money, we waste time. Every one of these patients is being poisoned because we know it's not going to work. It's just such a stupid system. And so in Kathleen said, when I started my career, it was very sexy, start a biotechnology company and be a big guy in private way. You're on magazines and they take pictures of you in lab coats and you're holding up. And the company was a success. We raised millions of dollars, we sold it for hundreds of millions of dollars, and then we still see cures going down, costs of medicines going up. I was part of that process, and you could tell a little story, an economic story. We created a great company in Canada, and sold it for a lot of money, and we were very famous. But then it's not really having a big impact, right? We we still make medicines that are unaffordable, we still have these diseases we can't cure, there has to be a better way. And so that's when we started to work on the Structural Genomics Consortium, which was an idea to, why don't we take money from companies, from governments, from foundations, all around the world, and decide to work on the proteins way down here, and our bargain for getting the money is to share the science with everybody in the world, to try and raise the amount of knowledge about the unknown. So it was founded in 2003, and now we have labs all around the world, including Pela. We never filed for patents. We believe that if you file for a patent, being secret causes lawyers to come in the way. It causes people to think that you're not being nice, and so we just say, no patenting, we'd always share everything. And despite the fact that we never patent, companies have given us $200 million in cash to do these experiments, right? Because they know, they want us to find the cure, the reason that Parkinson's disease, for example, is the problem. They can't do it. If they let the NIH and FAPESP do it, they'll study the same proteins over and over again. Nobody will study the unknown. So if everybody puts in a little bit of money to study the unknown, it's a good deal for everybody, and that we convince them. And then over the last years, I mean, just because we put things into the public domain, still we get a lot of papers. We publish 2.5 papers every week, 30% of them in the high. We've done 10% of all the human protein structures ever done. Many clinical trials have started from our work. You know, one of these proteins ends up being interesting, and then companies start to work on it, and molecules are being tested, and lots of companies have been. Okay, so we're doing open science, but then something struck me the other day. This is, remember the little baby I showed you at the beginning? So that's my son. That's his wife's sister, okay? When she was 4 years old, she was diagnosed with medulloblastoma, which is a brain tumor, and she was treated heavily with radiation, and that's a cure. But what happens is that because it damages your brain, you grow up stunted, your intellectual growth is stopped, and that was Carly last year. So you can see she lost her hair. She died about a year ago when her cancer came back, and when her cancer came back 24 years later, okay, her mom phones me, because I'm a scientist, right? Why are they offering Carly the same medicines that they gave her 24 years ago? You think? Right? All this time we publish our papers in Nature and we think we're famous, and then this happens. We're having no impact, right? And so I think this really touches home that we gotta work harder. And so, and it's nobody's fault, it's the system's fault, right? The system is stupid. So can we start sharing more and use the $300 billion to do more research per dollar? That's the hypothesis, okay? And so open science is a way that we can create businesses, because you have to do it in a business-friendly way, to try and make medicines under a different business model. And the strategy I use comes from salad dressing. So, Paul Newman is an actor. He's dead before you guys probably knew him, but he was a famous American actor that in 1982 decided to start a company to make salad dressing. And he started the company to give money away. So the company makes a good product. Newman's own salad dressing, we buy it, it's in our grocery store. After the company pays its employees, pays in research, pays taxes, the profit gets donated to Newman's Foundation, and they give it away. Let's give it all away. Since it began, it's given $535 million to students, to African children, to all around the world. And I thought, that is so cool, right? It's a business, it employs people, people are happy, it makes a good product, but the whole point is to give it away, not to make rich people richer. And so why can't we create a new drug discovery world like that? And so we did. So the first thing we did was we created, so the intention is to start from genes, which we're all molecular biologists, and end in affordable medicines. Not just medicines, affordable medicines. And so the idea is the first thing you need, like Newman's Foundation, we created a charity. called the Agora Open Science Trust. And that charity is overseen by 5 people. It's a Canadian charity, 3 Canadians. Michael Morgan, who was the principal scientist who led the Human Genome Project for Wellcome Trust. So one of the big movements, that's probably one of the grandest scientific projects of the last century, and he was in charge. And we're getting another person, she's not quite agreed, so I don't want to say her name publicly, but it's someone from a Geneva organization that's very well known. And so she's and us are going to, if there's any money, and there may never be any money, we'll choose to give it away. And it's not coming back to us, it's not coming back to any of these companies, it's not coming back to SGC, we're giving it away. And if it's bed nets in Indonesia, we'll give it away. Okay? We'll decide. And then we thought, now we set up under that companies that are for profit, just like Newman's Own, right? Just like the salad dressing company, it makes a profit, it employs people, it doesn't lose money. And so these companies are drug discovery companies on this side and service companies on this side. Companies that sell, like, can you make this protein for me? Sure, give me $100, I'll give you the protein. I make 5% extra money, $5 goes up to here. Those are service companies that work on a fee for service. So we imagine 2 kinds of companies. This company's already been formed, this company's been formed, this company's been formed, and at the end of the day, I'll tell you about, I want to do one of these in Brazil. Okay, so Meds for Kids, M4K, Medicines for Children, Meds for Kids Pharma, is the first one we did, and I'll tell you about it. It's 100% of the company is owned by Agora charity, right? Because, so the idea is if it invents a medicine, if, like in my old company, I started the company, I raised venture capital money for the first round, and then a year later I raised more venture capital, we raised maybe $40 million. Every one of those people who put their money into my company wants 10 times the money back. Yeah? And so as soon as you take money from a private investor, they want the most money back. Well, that means when the drug gets made, it's going to have to charge a lot of price because you have to pay all the people that you, right? So I thought, well, we can't take $1. Let's just take $1 from that. They say charge the highest price, charge high price. So we're going to keep the company 100% owned by the charity, whose mission is to charge as low a price as we possibly can. Okay? It's formed to invent new medicines, and I'll tell you about the disease. And it's the first company in the world ever to be done this way. So now I have to explain a little bit, because the business model is very confusing, because everybody thinks, well, where are you going to get the money? So let's just remind you what drug discovery is. The first step is, of course, you have to invent the drug, right? That scientist in the lab inventing an inhibitor or an antibody that in the animal does it. And so then, once you think you have a drug, and you've tested it in 2 mammals, like a dog, and it doesn't make them sick, and stuff, you go knock on the door, and you collect enough data to convince the regulatory authority, like the FDA, this is safe enough to put into a human being for phase 1 clinical trials. If it doesn't cause harm in the phase 1 trial and you get your dose right, then phase 2 trial is in the patients with the disease. That's the first time a patient with the disease takes it. Most drug programs go dead because it doesn't work. The hypothesis was wrong. And then you do, if that works, you do a phase 3 trial, which is a much larger trial in the patient population. And then, after the phase 3 trial is finished, you file a new drug application and you go to the regulators again with the data. Now, please give me permission to market my drug. Okay, so there's 2 regulatory steps, 2 steps where you have to knock on FDA's door and say, could you please let it go to people, and could you please allow me to get marketing approval, okay? These are formal legal processes, and the same thing in Brazil you have to do with your authorities, in Canada, Health Canada, in Europe, the European and EMA, you have to do it. So every place around the world has these regulatory, that their first job is to keep the citizens safe. The second job is to allow medicines and regulate medicines on the market. Let's put some money to this. approximately, and it's different for different diseases and etc. Approximately, to invent a preclinical candidate, the one that you think is ready to go into animals, can be 3 years, 7 years, $5, $10 million, depending on the complexity of the idea. The preclinical studies are the two mammal tox, the scale-up of the compound, the formulation, all the stuff that makes it a tablet or an injectable to go into human. That costs this much. The clinical trials are expensive. So the recruiting the patients is expensive. Paying for the patients to go, paying all the doctors, Okay, that's it. And then after the drug is approved, it still costs a lot of money. You have to have a factory, you have to sell people, you have to have a sales force, you have to do post-marketing, phase 4 as it were watching, did anybody get sick on my drug? Surveillance is called, and that's 10 of them. So it's a lot of money, okay? All right. So, if it costs a lot of money and people are putting money in, they want to know, how do I stop another person from coming in and selling my drug once it's approved? How do I keep competition away? What you've heard about most is patents, yeah? From the time you file your patent, okay, you have 20 years of protection. No one is allowed to use your invention you can exclude people from using your invention, okay? It protects against competitors' use of it, be it a compound, a method, a formulation, but the compound or the composition of matter patent is the strong one, okay? That's the important one. But guess what? In 1983, Hatch-Waxman signed a legal act in America that said, we want to encourage generic competition. And so we'll let generic firms file accelerated NDAs where they don't have to do all the clinical trials again. They can just use their drug and make sure it's the same as a Pfizer drug by comparing the data that Pfizer has and the data that the generic has, and if they're the same, they can get an accelerated NDA. But then we thought, well, that's too much for generics, they'll enter too quickly. So to balance that, they allowed the first company to hold the data, that NDA, remember, secret for a while, okay? So you get, currently, you get your patent to protect competition, but also, right, the data that you use to gain the approval is also secret. So you have two forms of protection against competitors. One, the patent, which it says you're not allowed to use this. Two, even if there is no patent, to get your drug on the market, you need to show your drug's the same as mine. You need to see my data, but you can't. In America for 5 years, 10 years in Europe, it's a long period of time where de facto you keep the competition away. Nothing to do with patents. You don't have to have a patent, you have the data. Because the way that the generics get on the market is to look at the data. If they can't see the data, they can't. Okay? There's another form of protection that this has been put, so the data, well, let me go back up because it's really confusing. This is why it's so complicated. No one started a company like this before. So you have the data, right? But a generic can say, well, I can go all the way back to the beginning again and pay the entire trial myself, and then I can go on the market. Yeah, so that's one way they can do it. And then they can compete. But what the American government tried to do to try and encourage more companies to go after neglected diseases or orphan, rare diseases, they passed the Orphan Drug Act that said, if you get your drug approved through that act, no one's allowed to sell that molecule even if you can look at the data. It's market exclusivity. So in that one, the FDA will say that chemical compound, that exact structure, only can get sold by Apple. No one else can do that. And that's marketing exclusivity here. So the market for which is granted, like America, it's granted after approval, and it protects against a competitor marketing any drug with the same medicinal ingredient. A lot of governments don't like this. There was a story, I can't remember the guy's name, it's like Shreckley or something like that. Thank you. And so what he did was he noticed, I think, that there was a drug on the market that had been being sold for 40 years, and it was pennies. But nobody had taken it through the formal regulatory process. So he thought, I'll pay for the clinical trials, the NDA, all that kind of stuff. I'll get orphan drug designation because it's an orphan disease or a tropical disease, I can't remember which one it was. And then when you get it approved formally, then this happens. So even though the drug was available for pennies, because he got this law, it's not patented, he could now, or that company could say, no one else is allowed to sell this in America except me. And then when he did, the price went up to $700 for pennies. And nobody has the legal right, because this is an act of Congress, right? That you can exclude competition. So most governments hate this, right? Because it's a way for companies to charge high prices. We're using it for a different purpose. And we can think that we can use this to charge as low price as we can. And so you say, well, how come nobody else does that? Well, in fact, people do. Some people have done it already. But let's show you the timeline of market protection. So this is after your drug is approved here, how much protection you get from competitors. This is a typical drug discovery program where you file your patent, and then science, clinical, IND, remember, investigational drug approval, clinical trials, you file for the new drug application. Here the regulators look at the data, and then they approve your drug. The time they looked at your data, you get it back. Okay, so on average, it's like 10, 12 years of protection. But look how much you get from the regulatory stuff. These are how much your data are seen before, and then you get additional time if it's a neglected disease, additional time if it's orphan, additional time if it's pediatric. It's a pretty long, compared to this. And so we thought, well, these you can get without a patent. You can get without a patent. And so therefore we can do drug discovery in the open, and as long as we get marketing approval, once it's approved, no one else can sell the drug except us. So it's a way that we can share the costs around the world to invent the medicine, and then guarantee to somebody that if you make this, if you manufacture and distribute this drug, you're the only person who's allowed to do it for 10 years, right? Because that's how long protection you have. So it's a good business incentive. If we can make the medicine, we give it to a manufacturer and say, you have exclusive right to sell this for 10 years, but you need to charge low prices. If we can make the medicine, we give it to a manufacturer and say, you have exclusive right to sell this for 10 years, but you need to charge low prices. And they'll say, okay, it's like free. I did zero research, I get a product to sell, and I have 10 years to sell it. That's the big plan. I have a rich fantasy life. That's my fantasy world. Okay? And so let's just look at this regulatory exclusivity and some of the details. They're like patents. They shield the product against competitors. They're consistent with open science. For a patent, if you talk about it, if I talk about something in here, I can't patent it anymore. Because I've disclosed it to the public, it's no longer covered. I've created prior art, and a patent has to be new. So I can't talk about it. So if I'm working in a patent world, I don't say anything, right? Open science, I can tell you all the science I'm doing. It's costless. Every year, if you have a patent and you want to protect your market in America and in Europe, every year you have to pay to keep that patent alive. And it's really expensive. This, you don't have to pay anything. It's not subject to challenge by competitors, right? So if I'm Pfizer and I invented a drug, and Kathleen's a generic company, and she goes, oh, I really want to sell that, she'll say, I'll sue Al. I'll say, that wasn't a good invention. You guys, it was obvious. It's not an inventive step. I want to overturn the patent. Yeah? It's a good strategy. So let's pretend after day one she overturns the patent. And then she says, now there's no patent, I can sell it. Uh-oh. I have to prove to the regulators that her drug is safe. To do that, she has to do this NDA bit. To do that, she needs to see my data, and she can't see it. So even if the patent is overturned, my data protection wins, right? And she can't challenge it. So regulatory exclusivity is not subject to challenge. She has no choice. She has to wait 5 years, and then she can enter the market. And it provides a period of market protection. This was written by my lawyer. I had no clue what that said. But so that means in advance. Let's pretend we're doing an Alzheimer's program and I patent on day one. I don't know how long the clinical trial is going to take. I don't know what my patent life is going to be at the end. 20 years starts. Tick, 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 tick. I'm doing my clinical trials, and my patent life is going down, and I don't know how long it's going to be. But with regulatory exclusivity, even if it takes 30 years, once it's approved and there's no patent life left, I still have the regulatory, I still can keep competitors away. Okay? So it's certain in advance, right? Does this have any influence in the market, in the business world? Absolutely. The Orphan Drug Act, remember when it was passed, it gave people marketing exclusivity. If you look at it, didn't affect patents at all. It had nothing to do with patents. They just said you get exclusivity in the market, like I described before. If you look at the graph, it goes, nobody's researching orphan drugs, Orphan Drug Act, people voted with their pocketbooks. They said, now it's worth it. I'm going to go and invest in orphan drugs. Okay? Had no effect on patents, but it affected the market. I think we counted 27 drugs that are being sold that in the Orange Book, which counts all the patents behind the drugs, that have no patents. So there are already drugs on the market that have been approved where there's no patent life, and it's the exclusivity that's keeping them. And these are new drugs. These aren't old ones. And then finally, is one from Canada. Do you remember Trump wants to renegotiate the NAFTA agreement with North American Free Trade Agreement with Canada? And in trade agreements, they fight over everything. And always the last two things is agriculture and drugs. And the last thing that was approved in this deal was regulatory exclusivity. America, which has all the companies that make biologics, they wanted 2 extra years of exclusivity to help their companies. Canada, which that means we have to pay high prices for 2 other years, we went no. It was the last thing among the last things negotiated. So regulatory exclusivity is so powerful that when countries negotiate with each other, it's one of the last things they can agree on. That and milk price. So we said, let's form M4K, and we say, forget about patents. We'll focus entirely on regulatory exclusivity as our business asset, okay? And so then, because we can do open science and we share our data, governments can fund us because we're really doing science, right? We're testing the hypothesis that inhibiting this protein will cure cancer. It's a hypothesis. We treat it exactly like a science experiment. So it uses open science. We do no patents. We put our chemistry online every month. We have a webinar, internal chemistry meeting where we're showing compounds that, if you're a pharmaceutical company, you never show those because it ruins your patent life. For us, it ruins everyone else's ability to patent, right? Then we post them on YouTube, so you guys can go watch. If you want to see our drug discovery meetings, you can log into our webinar. It's pretty boring. It's a lot of chemistry. And then we put even data in open lab notebooks where the postdocs every month type up their experiment, put it online for the world to see. The regulatory data, we'll keep it behind a firewall or whatever, I don't know what the right word is. You have to click to be able to see the regulatory data, and then all you do is you click, it says use it for anything you want, but don't use it for regulatory purposes. Okay, so the last thing we want is for us to invent a drug and intend to price it low, and evil Caitlin comes in and says, I'm going to use Al's drug, Al's data, and race him to the market and get approved first. Then I can stop Al. from selling this drug, and I'm going to charge high prices. Okay, so that's the only thing. If somebody looks at our data and says, Al, you're stupid, you should use a methyl group there, that's okay. And they make a new molecule and a new invention, that's okay. But just don't use our exact molecule and our data and beat us to the market. We don't think that's fair because it'll lead to high prices. And so the idea is to, this is written by my lawyer. It's all big words, let's forget it. But the bottom line, it's wholly owned by Agora, right? Which is dedicated to furthering the public good through open science, and any licensing revenues to M4K will be given to Agora to give away to the world. That's the model. So let's contrast this business with the other businesses that I've founded, which are traditional ones that you hear about. You hear all these biotech companies in Boston and stuff. They do this, right? The company's not owned by a charity. It's owned by either public investors through when you go on NASDAQ or the stock exchange, or private investors, and they're investing in medicine to make money, right? The medicine is a product. They don't think medicine is a human right. Medicine is the product I'm investing to make money, okay? Then they say, if I'm putting my money in, I want to keep everybody away. So they patent, they license it with lawyer agreements, they put and they get regulatory exclusivity. They can raise money from venture capital, from the equity markets, like the public markets. They can generate money. They can also get grants and foundations, and they're very protective of their intellectual property. That's how I ran my company before, many of them. But in this one, there's no shareholders. Agora owns everything. The assets to keep people away are the same, except we just don't have patents. We are unlikely to get venture capital or internal cash flow because we're going to give everything away. But we're more likely to get grants, foundations, and philanthropy in the open model. Remember, around the world, there's $150 billion spent on grants and foundations. The entire world's venture capital in biotech is $10 billion. So I have $150 billion to compete for in my company, and I'm giving away 10. Doesn't matter. Okay? And here we're uncomfortably open. Instead of fact piling, we put everything, we share our results all the time. So the first disease we're going for is called this one. It's diffuse intrinsic pontine glioma. Glioma is a cancer of the brain. Diffuse means that once you found it, it's all over the brain. These are the lifespans of the common pediatric cancers, and there's the IPG down there. Luckily, it's extremely rare. But on the other hand, that means there's no business incentive to do research in DIPG. And there was this young lady who since died. She started this lemon face challenge where people that suck on lemons and gave money to charity for her. So in 2014, when they had new biopsy methods, they could sequence the DNA of these tumors, and they found out they were genetically really kind of prescribed. A lot of them, most of them have an histone H3.3 mutation, and it's usually lysine 27 to methionine. This is an epigenetically modified amino acid, so there's something to do with epigenetics. And then on this background, you have various other mutations, and about 25 to 33% of the patients have an activating mutation in this kinase. So the mutation turns the kinase on. Okay? So for a drug discoverer, a kinase that's always on is a good drug target, because then an inhibitor is the right approach. Fortunately, our lab at Oxford had already solved the structure of this kinase, had already had early chemical matter, had actually done some experiments in another system. So we said, this is a great project to start at M4K. We're going to make a drug. We're targeting this kinase and test the hypothesis that if we treat the children, their tumors will slow or go away. It's a hypothesis because nobody knows why this kinase turns the cancer on, but we'll discover that in our experiments, okay? And the first thing to notice that we have inhibitors already. They're not ready to be drugs, but we use those inhibitors in animal models and in cell-based models to show that the cells die when you inhibit this pathway. Okay? So that gave us comfort that there may be a drug to be made. And these have been published. We have CNS penetrant compounds. So these are our starting points for drug discovery. Inhibitors that work in animals, but they're definitely not drug-like. They're non-selective, and their pharmacology is wrong. So what we did, just like, now if we were in a biotech company, this is the kind of stuff we'd be doing. It's called a target product profile. That's what we're aiming for. And so this says enzyme potency, ADME, cell permeability, CYP inhibition, hERG, which is ion channel inhibition, and it says it has to be less than 10 micromolar, more than 500 micromolar. So we're trying to make a chemical that meets all of these criteria, and that's the one that we will try and advance to make a medicine. So every drug discovery project usually starts this way, by deciding what am I doing? Okay, that's what I'm aiming for. And so we have chemical synthesis, then we go to biochemical assays, then they go cell-based assays. We want to inhibit ALK2, but we don't want to inhibit its cousins ALK1, ALK5, and ALK4. So we screen them against one and the other to make sure it hits this one. It inhibits ALK2, but not these kinases. And then we start doing our animal studies and our in vitro studies, and then we profile it widely to make sure it's not hitting another kinase. It's kind of what happens every day at Merck or at Pfizer or biotech companies. That we're doing it in the open is just the difference, okay? This is very traditional. What's not traditional is I'm showing you chemical structures from active drug discovery projects. Now this slide would never be shown if I was Pfizer or Merck or a biotech, because that would be my intellectual property, okay? So these are some of the compounds that we made. M4K 2009 looks to be good. If it's green, that meets that target product profile. Remember right? And if it's yellow, it's kind of, but it's not really so bad, right? So this is mouse pharmacokinetics, how much the ADME properties, potency and selectivity, that's the off target. And so this is the kind of way that every month we look at this. Are we getting better? Are we worse? Let's put put a amide here, let's put a methyl here, test it. We make about a couple hundred compounds and everyone's getting a little bit better. And so if you take a step back, so the first program we started with this ALK2, we got $2.5 million in funding to do this already from the Ontario government, from charities, from the Canadian government. It's involving institutions around the world. We have companies that are donating stuff. These are two contract research organizations, Charles River Labs in Cambridge, England, Reaction Biology in New Jersey. They think it's so cool, right? So they're donating their time to the project because nobody's making money, right? If we were making money, they'd never do this. They'd say you get paid, right? But because we're going to give away the money, they, I love it. So Charles River, every one of their employees gets 1 day a month to donate to a charity. So the whole chemistry group said, let's donate all of our time to M4K. And we have a project team inside this lab in Cambridge, England, working, making molecules for us. They're awesome, right? They also just had a big dinner event where they raised $2,500 and they just donated it to us. It's so neat. The idea is really resonant. We have other universities saying, hey, can I help? I've got an assay system. Okay, I'll do an assay for you for free. If I'm a biotech company, I have to pay every single one of these people, negotiate for 6 months. You may not tell my contract, I own all the data. Oh, that's not fair. And it goes back and forth, lawyer. And really, these children are dying, right? We're talking about lawyers, who's going to own this stuff, right? And so the open model makes this happen like that. Even Boehringer Ingelheim, not even. Boehringer Ingelheim phoned us at one meeting and said, we've worked on this protein before, let us tell you what we learned. And it's amazing, a pharmaceutical company phones us and said, let me help you. Because they understand the business model, it's not competing with them, and they told us all sorts of information that was really helpful for drug discovery. And there's lots of doctors that want to help as well. And we've started animal tests, and we should hear in 2 weeks how the animal tests went on our molecule. And you can Go on YouTube and you can see everything. It's really boring. But if you want, you can talk about a bunch of chemistry nerds talking chemistry. If you're interested, the details of all the business and the lawyer stuff has been published. We published it in Welcome Open Research, so everyone can get it for free. Oh, I had to make a little joke. So, I was born in Wales, right? And so my name is Aled, which is a uniquely Welsh name, Morgan, which is a Welsh name, and Edwards, which is a Welsh name. Max in the lab, his last name is Morgan. And Max, both of these names are common in Wales, so we share a name. Owen was the founder of that, helping found that company that I told you that we sold stuff. And he's Welsh also, and he shares a name with it. So we just thought Wales is a really small country. Oh, we have 3 people. We're very excited. So that's why, because normally I go Aled M. Edwards, right? Owen G. Roberts. But in this one, we spelled out all the names so we could have our little Welsh party. So on the basis of the success in quotation marks, now true success is of course making a medicine, right? But to prove the model successful, we need to try it on other areas. So we created M4NB, neurodegeneration or neurological, whatever we haven't decided. Yeah, NB. Same deal, 100% owned by this. We have 2 drug discovery programs that are really early that we're starting. And we got $1.5 million donation from a donor, from a philanthropist in Canada, who wants to say, test the model. I'm interested to see if this research and business model work for neurosciences. And that's kind of cool. So we started this project already. It's structure guided. It's occurring at McGill University. We also got money from the Michael J. Fox Foundation. And so, is this an opportunity for Brazil? Your healthcare, you're not a rich country, right? You have 200 million people, Canada only has 35, right? How do we make medicines that are for new medicines that are available to everybody? So, can we start the concept of we use this open business model and apply it to the diseases that are important here? Can we do the exact same thing? Create M4ID, medicines for infectious disease, have it owned by a charity so it tells the world this is for the world, it's not for me to drive an expensive car or buy a helicopter or something. Start with diseases that matter, that are difficult for the normal business model to work. Commit to open science, commit to no patenting, commit to sharing. It will attract money. I've seen it in my other lives, right? We already started great collaborations with Pharma, the Gates Foundation, the NDI. A whole bunch of people are already doing science, kind of in the open already, but just not formally under this structure. SGC Unicamp can be the leader of this one and say, let's just do it and start. And then if we can take some of these projects that look exciting in our malaria project or our leishmaniasis project, why don't we start and create a company owned by charity to take this all the way through? And I think it's really possible. I think if you go out, I spent some time in England and they always said, infectious disease is really hard, the business model doesn't work, we need somebody to do something. Then you go to America to the Pew Foundation. Oh, the business model's broken. We need a dramatic, and then they don't do anything. They just do the same thing over again. But they talk. Oh, we need a sea change. We need a game change. So why don't we do it? Canada has nothing to lose. We don't have any pharmaceutical companies. You guys don't have any big pharmaceutical companies, right? Why not do something completely different? You're free. You're unshackled, as it were. And so I think we do it, and for those, there's many people from SGC here, but for those who don't know what the SGC is, it's one lab of 6 labs around the world. There's about 350 scientists around the world. This is a lab here. It's up at the top of the hill in Lactat. It's got lots of collaborators from all around the world, the large pharmaceutical companies. We have Gates Foundation, the Wellcome Trust. Everybody wants to make this a success, and we've decided we're going to have the biggest impact in infectious disease. We have lots of projects, but I think that's where we can be globally unique here, not just unique in Brazil or unique globally unique. And I think it's pretty exciting, and I really like to try, right? I hope it'll be fun trying, die trying, that's the word. Okay, thank you very much. I'll repeat the question.
And I agree with you. A lot of big companies make this very, a little bit more free. They don't have any much bother us. But then my question is the other side, because we don't have any big pharma companies here. We have lots of science in academia, but not really. So it's really, the science is really below, inside the university. You think is the fact they don't have big companies, is that the business model is not that fresh. I think this could be a real drawback in developing more equipment.
Yeah, so the question was that, unlike other countries, you don't have any big pharmaceutical companies. The research is in academia and there's nothing in the middle. Can we develop a new ecosystem to make medicines with nothing's in the middle? Very little experience in medicinal chemistry, very little experience in preclinical toxicology and stuff. Well, that's where we can help, right? In the SGC, we have 3 or 4 scientists who made drugs before. They come from pharma, now they work for us. Stephen Fry is very keen on this. He's not in the SGC, but he's a friend who loves the idea. He used to head up chemistry for Glaxo. Chaz Boucher, who heads up our lab in Oxford, used to be head of neuroscience biology at GlaxoSmithKline. So in the SGC, we've done it before, and it's not impossible to recruit people here. So it will be a challenge for sure. But the advantage is we're not here rigorously. You know, we don't have 100 biotech companies going, stupid idea, we need more patents, right? We have 0 biotech companies, right? So it's green, brown field, as it were, we can build.
Because your model is basically less greedy than all the other big pharma companies, do you have a problem with them buying your talent?
So absolutely not. I mean, I don't fault the pharmaceutical companies at all. We created a capitalist system. We as a society have decided that medicines are a product. All of us probably, or I don't know, the older ones probably have stock somewhere in a pharmaceutical company. We could always go to Merck's annual general meeting and say, make less money, but we don't, right? And so rather than demonize the system, everybody I know in the pharma industry and the research side is trying to make a difference. They go there to try to help. The business model is the problem, not the pharma, and we're all part of the business model. So we have 2 choices. We can throw arrows at them, but I don't think that's the right thing to do. Create another universe. That's a much more positive way of doing it. And their purchase, if I was the CEO of Merck, I'd probably do the exact same thing. No, it's my fiduciary responsibility to make as much money as I can, right? That's how the world is interpreted now. We may disagree on that, but that's how it's interpreted. And so how can we begrudge them trying to make as money as they can within the law, right? And so it's a delicate question, right? Because sometimes they do things that are illegal, but they get fined for it. I think it's more productive to create something that works rather than to try and, dismiss something that doesn't.
Sure, but do you have any sort of training to those companies of people who any difficulty recruiting people that you guys are really interested in?
Our pharmaceutical partners benefit from lots of people who are trained by us. But the good thing, looking around the room, is there's always brilliant young people to come in on the other side. And it's our job as academics to train people for their lives, right? So we think it's a success if any of our scientists are recruited, right? We're contributing to the economy and we're providing new opportunities to train young people at the same time.
short and examination how you can do the discovery. Just want to ask, do you have any platform for the screening of like compounds? Because there are a number of academies doing research, making molecules, but they don't know how to screen molecules. There are like many synthetic chemists screening crazy molecule, but they don't screen for any disease sometimes, and they just publish, okay, this is a new synthetic way to synthesize a compound. So do you have any screening platform where people can contact you and then, okay, we have these molecules, we have so number of molecules, can you please screen it for this disease? So,
Yeah. So it's a chemist who doesn't think that his or her compound is the next cure to everything. Okay? The odds are that we screen thousands and millions of compounds and we can find 4. The probability that any chemist in the planet has made a compound that is going to work is 0. And so it's incredibly time consuming to talk to every chemist and screen their compounds for them. That said, if there's a hypothesis-based generation of the compound, like there's a scaffold that we know binds, and we have many platforms at the SGC that can screen compounds. We have many assay formats, we have many different proteins purified. So we can do that and other people can do that. Of course, with the caveat that the chemist has to be more than I made this molecule. It has to be made with an idea that it will interact, test a hypothesis that should be tested. Because there's so many compounds out there, right? It's really difficult to know which one is the right one. And the odds are that any chemist made the ones. Okay, cool.
Those pharma partners that you pointed there, they contribute to your research much more with information on your own funds.
So the question was, these pharma partners down here, AbbVie, Pfizer, how do they contribute to us? They contribute with funding. So each pharma partner gives about American dollars, $5.5 million to join for 5 years. They also participate in about 4 to 5 collaborative projects where, let's say with AbbVie, the chemistry would be done in AbbVie, and together we try to make a new inhibitor of a protein to give away. And it's an early inhibitor, but by giving it away, then every professor or every student can use that compound in their assay to see if it works. And so it's far more than the money, which is important because we need that. The ability to interact with expert medicinal chemists and drug discoverers in each of these companies is as, priceless, as it were. And
Can I add something about the graphing?
yeah.
Yeah. So here, Unicamp for our project, right? Because our SGC is a national innovation unit from Embrapa, Empresa Brasileira de Inovação Industrial. When we make a contract with a pharmaceutical company, so nowadays, Pharma, they give us cash to run a project with them, which has to be innovation, not service. And this Brazilian institution matches funds in cash. So that is a model that is working in Brazil, and how we are able to actually attract Brazilian pharmaceutical companies to give us cash for a project. It's not that we'll do it for them, we'll do it with them. For example, there is people in this room who are postdocs from the pharmaceutical company working together with us. So we are able to pay people, reagents, and they want to accelerate drug discovery.
So to fact this question before, in a collaboration with Ashe, we're working on a kinase down there. They're doing the chemistry. I think, how many papers? We published 1, and now the second paper.
Yeah.
So it's an Ashe chemist, the Unicamp scientists collaborating on a new chemical inhibitor of a kinase that no one studied before. A new compound, a new structure, a new idea, no patent in the public domain. Ashe, their scientists learn, to the extent that there's not a lot, so they're learning. We're benefiting from their chemistry. It's a really cool collaboration, all under open science. And so it's happening in Brazil, and Europharma is on different projects is the same. it's really changing, I think, the way in which these companies are going to interact with the academic world to try and, as you say, create a closer link so we can go all the way from the gene